Welcome to this lecture on the synthesis of ribosomal RNA, or rRNA. Imagine a massive car factory that needs to build thousands of cars every single day to meet global demand. Before it can build a single car, the factory first needs to build the actual robotic assembly machines that will put the cars together on the conveyor belt. In our bodies, the cell is that bustling factory, and the proteins are the final cars being produced. Today, we are going to learn exactly how the cell manufactures the structural parts of these robotic machines, a process that is absolutely essential for the cell to function and survive. Now that we have our factory analogy in mind, let us look at the biological equivalents. The robotic assembly machines in our cells are called ribosomes. They are responsible for piecing together amino acids to build proteins. The synthesis of rRNA is simply the biological process of manufacturing the main structural components of these ribosomes. Because proteins do almost all the active work in our bodies, from digesting food to fighting off viruses, the cell must constantly manufacture millions of ribosomes to keep up with the demand. Before a cell can do anything else, it must build these machines, making rRNA synthesis one of the most urgent and continuous tasks in all of biology. Understanding the urgency of this process, let us dive into where and how it actually happens. In eukaryotic cells, rRNA synthesis takes place in a specialized, dense region of the nucleus called the nucleolus. You can think of the nucleolus as the dedicated factory floor for ribosome production. Here, a highly specialized enzyme called RNA polymerase I goes to work. It reads the specific genes on the ribosomal DNA and transcribes them into a massive precursor molecule known as the 45S pre-RNA. This giant transcript contains all the core RNA components needed, but it is not quite ready to be used yet. It must undergo further processing to become functional. Since the 45S precursor is too large and unrefined, let us see how the cell processes it. This giant precursor molecule is carefully chopped up and chemically modified by various cellular tools. This cleavage process yields 3 smaller, mature rRNA molecules, specifically the 18S, 5.8S, and 28S rRNAs. Once these mature pieces are generated, they do not just float around aimlessly. They will eventually fold into highly complex 3-dimensional shapes. Finally, they combine with specific ribosomal proteins to form the functional large and small subunits of the ribosome, ready to be exported to the cytoplasm to start building proteins. Now that we have discussed the steps, let us visualize this entire process. In this diagram, look at the top horizontal line representing the ribosomal DNA. You can see multiple blue circles, which are the RNA polymerase I enzymes, moving along the DNA. As they move, they synthesize the long, wavy green strands of the 45S pre-RNA precursor. Notice how the strands get longer as the enzyme progresses. In the lower half of the diagram, we see the processing stage. The single long 45S precursor is cleaved, indicated by the downward arrows, into 3 distinct, separate segments. These are the mature 18S, 5.8S, and 28S RNAs, perfectly cut and ready to form the ribosome. Having seen how rRNA is made, we must ask why it commands so much of the cell's energy. The reality is staggering. Ribosomal RNA accounts for about 80% of all the RNA found in a typical eukaryotic cell. Because the cell relies entirely on proteins for survival, any disruption is catastrophic. If a toxin or a genetic mutation halts our RNA synthesis, the cell rapidly loses its ability to make proteins and will undergo cell death. Medical science takes advantage of this vulnerability. In fact, many modern cancer therapies specifically target rRNA synthesis because cancer cells desperately need to build millions of ribosomes to fuel their rapid, uncontrolled growth. To truly appreciate the massive scale of this biological manufacturing process, let us look at its mathematical representation. Biologists use a simple linear equation to model how fast a cell is growing. The formula is N total equals G times RTX times T. Here, N total represents the total number of RNA transcripts produced by the cell. G stands for the number of active RNA gene copies present in the DNA. RTX represents the rate of transcription, meaning how many transcripts one gene produces per minute. Finally, T is the total time elapsed in minutes. If a cell needs to boost production, it physically must either activate more genes, increasing G, or speed up the enzymes, increasing RTX. Now that we have our formula, let us apply it to a concrete example. Imagine a rapidly dividing human cell. We are given that it has exactly 400 active copies of the rRNA gene, a transcription rate of 30 transcripts per minute, and a time of 60 minutes. We want to find the total production. We use our formula, N total equals G times RTX times T. We substitute our values, 400 times 30 times 60. Multiplying the genes by the rate gives 12,000 transcripts per minute for the whole cell. Multiplying that by 60 minutes gives us our final answer. The cell produces a staggering 720,000 transcripts in just 1 hour, highlighting the incredible efficiency of the nucleolus. Having successfully used the production formula, we sometimes face a different problem: predicting cellular timelines. Suppose we know how many ribosomes we need, and we want to find out how long it will take to make them. To do this, we must derive a new expression from our base equation. We start with N total equals G times RTX times T. Our goal here is to isolate the time variable, T, on one side of the equation. As our first mathematical step, we divide both sides of the equation by G, the number of active genes. This algebraic manipulation begins to separate the time variable from the cell's genetic resources. Continuing from our previous step, we now need to completely isolate the time variable. We currently have N total divided by G equals RTX times T. To get T by itself, we divide both sides of the equation by the transcription rate, RTX. This gives us our final rearranged formula. T equals N total divided by the product of G and RTX. Intuitively, this makes perfect sense. The total time required is simply the target number of transcripts divided by the cell's overall production speed per minute. It is exactly like calculating how long a road trip will take by dividing the total distance by your driving speed. Now that we have proven our time formula, let us see this with a concrete example involving cell division. A yeast cell needs to manufacture 3 million new ribosomes to divide. We are given a target N total of 3 million, 150 active genes, and a rate of 20 transcripts per minute. We use our derived formula, T equals N total divided by G times RTX. Substituting the known values, we get 3 million divided by the product of 150 and 20. The denominator becomes 3,000, meaning the cell produces 3,000 transcripts every minute. Dividing 3 million by 3,000 gives a final answer of 1,000 minutes. This translates to roughly 16.6 hours,
proving mathematically that ribosome synthesis is a major time-limiting factor in the cell cycle.
We have covered a lot of ground today, so let us reinforce the most important ideas from this topic. First, remember that rRNA synthesis is the absolute foundational step in building the cell's protein-making machinery. Without it, the cell cannot survive. Second, this high-volume manufacturing occurs in the nucleolus, where RNA polymerase I transcribes genes into a massive 45S precursor. Third, this precursor must be precisely cleaved into the mature 18S, 5.8S, and 28S components to function. Finally, we saw how this production can be modeled mathematically, proving that cellular growth is strictly limited by how fast it can build these factories. You now have a complete understanding of RNA synthesis. Great job!